then the first one we'll cover for today's lecture online is going to be proteins. So when you think about proteins, this image right here may be really familiar. You'll think about things such as eggs, definitely your meats, your eggs, your nuts. So we're going to talk all about proteins. And it's not just about food. Proteins are pretty prevalent in our lives in biology, and we'll talk about some examples where you see them. All right, so one of the characteristics of a protein is it's typically comprised of those elements that we talked about before. So carbon, hydrogen, oxygen, and nitrogen, and a lot of other elements, especially sulfur, and there can be a few other ones. So we'll talk a bit about what the names are for proteins as a polymer and macromolecule, as well as the monomer names. You're going to see proteins in every aspect of life. So when you think of movement, your muscles are comprised mostly of protein. Your metabolism, as we'll talk about later, they're going to be proteins called enzymes that are really critical as our cells communicate with one another, and either the structure of our body is comprised of proteins. So this is a really critical thing to understand, which is the monomer of a protein is going to be called an amino acid. So an amino acid is really, really critical for you to understand. So remember, the monomer is just a single unit. So the single unit of a protein is an amino acid. An amino acid has a very particular structure. The first thing is a carbon that's going to be in the middle. So you have a carbon in the middle, and if you remember about carbon, carbon's going to have four areas where it can have a bond. The area with the nitrogen group is called an amino group. So every single amino acid will have these components. Number one is an amino group. Number two is a carboxyl group. That's another carbon and oxygen and hydroxyl group. And then the final area is this special space called an R chain. So every single amino acid is going to have an amino group, a carboxyl group, and a side chain or R chain. And the really critical component of an amino acid is going to be this R chain. So this is where you have some variability. Every single amino acid are going to have amino groups and carboxyl groups. You don't have to memorize what they look like, just being familiar that amino acids are made up of these things, but it's that R group that's going to allow it to function differently. So with these amino acids, like I mentioned before, the side chain or that R group is going to help determine the property of that amino acid. So some amino acids can be polar, some can be nonpolar, and some can be charged, and all of that is related to that that R group or that side chain that you have present. So this is just a picture here showing that the chains or the charges can determine how our amino acid is going to function. So some amino acids are nonpolar. If you remember that, that's no type of pull on electrons. They're going to be pretty even. Some amino acids are polar, meaning they're going to pull the electrons. And then finally, some amino acids can be acidic or basic. So the bond, the special bond that ties together or connects amino acids, is called a peptide bond. So being able to identify that, let's say for multiple choice, is going to be really useful. And this is where you have bonds between the carboxyl group and the amino group, not the R group. So if you're looking at amino acid chain, and you have a string of them along an axis, the thing sticking out would be the R groups, but the areas that are connected to each other are the carboxyl groups and the amino groups, and they're going to be connected by that peptide bond. A polypeptide is the name of a polymer for a protein. So remember, our amino acid is our monomer for our protein, and our polypeptide is the polymer for our protein. So this is essentially just a linear chain of peptides. So as you string together amino acids one by one and you form a line, that is going to be what a polypeptide is. The ends of the chain of the polypeptide are going to be free. So one end is going to have the amino group hanging off. Do you remember the structure here? The amino group. So one side that has the amino group hanging off is going to be what we called an N-terminus. An N-terminus. And then you'll have a chain of the amino acids linked together with the R group sticking out. And then the other side of that polypeptide is going to be called a carboxyl group, or our C terminus. So just being able to be aware that if we say N terminus, we're talking about the side with the amino group. C terminus is the side with the carboxyl group. So you may have heard this saying before, structure leads to function. This is a picture of an amino acid, and this is really key for helping us to understand that whole concept of structure leading to function. So proteins are organized in a very specific way. Initially, we start off with primary structure. Then we move on to secondary structure, which you see here, the purple and the teal, and then tertiary structure, and finally quaternary structure. So we'll go through each of these individually. So primary structure is essentially just having those amino acids lined up. So I have this in an additional piece beside your notes, but it's the very first step in processing. So as you're making your amino acids, and you have your chain that says we're going to have meth and pro and ti and lu and histine, all these things. As we know which ones to make, that is called your primary structure. So for instance, a necklace that's made up of a string of beads, that will be the primary structure. So this is just literally a string of amino acids. So I normally get to do a paper example. I can't do that right now. But a single sheet of paper, if you look at, let's say, the edge of that single sheet of paper, that will be your amino acid. So looking just at the edge of them lined up, straight line, that's going to be your primary structure. Your secondary structure is where your, where your primary structure begins to fold on itself and create bonds. So there's two different types of bonds that we'll talk about. One is called an alpha helix. And if you've ever seen, let's say, a string of curly hair or a curly fry, how it starts to form that helix shape, this is going to be formed through hydrogen bonds, and it's called an alpha helix. The other type of shape is called a beta, so the B stands for beta, a beta pleated sheet, and that's going to be a zigzag pattern. So kind of like an accordion if you think of that. So secondary structure is once you take that string of beads or strings of amino acids and you either form a coil or you make a beta pleated sheet, that is going to be your secondary structure. This is really critical for structural strength of proteins. So again, if I had that piece of paper, what I would be doing with it was making it like an accordion style so you can actually see the bonds that you have in place. All right, the final one is tertiary structure. So with our tertiary structure, basically what you have is, of course, your primary structure is just a string. Your secondary structure is either a beta pleated sheet or a coil. And your tertiary structure is where you take all of that and kind of ball it up together. So if you look within the tertiary structure, you're going to basically see all of the R groups that bond together. So the tertiary structure creates this big, large 3D shape that is formed through those R groups interacting. Within the tertiary structure, you're going to see regions, that says of proteins, and these are called domains. So if you think of a balled up piece of paper, a little nook within that balled up sheet of paper might be referred to as a domain. And some proteins end right here. So this can be the final stage of a protein, although it's not always the final stage of a protein. So again, like that sheet of paper, this would be our tertiary structure. If you were to open that up, you would see some of the beta pleated sheets, you would see some of the alpha helices. And let's say we're talking about, one little teeny tiny dot in the middle of this. Let's say we want to talk about that zone or something. That could be referred to as a domain. So this is what tertiary structure looks like. And some proteins can function like this completely fine. Quaternary structure, this is where we assemble two or more of these tertiary units, so that ball of paper, two or more of those together, or two or more separate polypeptides. Remember, polypeptides are the name of the primary structure, and that begins to fold on itself. So two balls of paper, two of those different polypeptides come together. And then each portion of that big complex can be called a protein subunit. So a great example is your hemoglobin, which is used to carry oxygen, right? You can carry oxygen in your hemoglobin, or things like antibodies, which are made up of different portions. So again, quaternary structure would look like this. You have three different tertiary structures together, forming a big complex. And each of those areas are called protein subunits. So again, just showing kind of start to finish, your primary structure is your string of amino acids, no types of interactions. The secondary structure is where you start to form some interactions, so either a coil, which you see here, or maybe a beta-pleated sheet. Once those all come together, you form what looks more like a complex in your tertiary structure, and then multiple tertiary structures together give you a quaternary structure. So that summarizes proteins, gives you kind of a gist of it, and we'll talk about some more. The final macromolecule we'll discuss are nucleic acids. So nucleic acids, like one of our principles of living things, every living organism is going to have genetic material, and this is essentially the recipe for how to create that organism. Genetic material we'll talk about today are different types of nucleic acids. The two major polymers of genetic materials are DNA, which is deoxyribonucleic acid. See the D, N, and the A. And then RNA, which is ribonucleic acid. And I'll talk a little bit about the structure differences so you can figure out how they are different. So a monomer, remember we're talking about, this whole section is about macromonomers. So our polymer, again, like I said, is the DNA or the RNA itself, but the monomer, the individual monomer of a nucleic acid is called a nucleotide. So it is made up of three components: a phosphate group, which you see here in yellow, a 5-carbon sugar, pink, and nitrogen-containing base, or sometimes you see nitrogenous base in green. So we're not going to talk too much about the phosphate group, but with that 5-carbon sugar, we can either have ribose or deoxyribose, and we'll talk about them. And then with our nitrogen-containing base, there are essentially 5 bases we'll discuss: adenine, thymine, and uracil, and then also cytosine and guanine. So I'll tell you how they pair up in just a second. So with that 5-carbon sugar, again, ribose, deoxyribose, the major difference is outlined in red. So if you're looking at the bases of ribose and deoxyribose, you see that with your deoxyribose, you are missing an oxygen, so deoxy without oxy. You're missing that oxygen at the 2-carbon position, and ribose, you're not missing it at all. So this is where the difference in the name comes from. The nitrogenous bases, you see some of the nitrogenous bases here, and they actually pair up with each other. So I'll just show you that in the next slide and make it a little bit easier. So when we form our nucleic acid polymer, or when we form our DNA or RNA, our nucleotides are going to be linked together through this sugar phosphate backbone. DNA runs in one direction, and we'll talk about it very intently and in depth as we get to the last module for the course, but DNA is going to run from 5' to 3'. It always runs in that particular direction. So when we're looking at the bond between the nucleotides, that bond is called a phosphodiester bond. So again, just being able to recognize that word will be really great. And between the bases, you're going to see hydrogen bonds. Remember, hydrogen bonds are, material you'll see on your test, and those are going to be those weak bonds that you see between hydrogens and electronegative elements. And the combination for how they pair is A always binds with T or U. I'll tell you the situations in which it does bind with T or U. And G always binds with C. So some characteristics about DNA. Again, we talked about it being a deoxyribose. DNA is a double helix. So when you look at it zoomed in, you actually see 2 lines, and those 2 lines are then wrapped around each other in a double helix type of fashion. And in DNA, you have a T present, but you do not have a U present. So adenine only binds with thymine, and guanine only binds with cytosine. It's A and T and G and C. So DNA does not have uracil present, it only has thymine. So something really important to keep in mind. With RNA, really similar, but a few differences. It's made up of ribose, of course. It is single-stranded. So with RNA, you only have one strand. You don't have a double helix. And in RNA, you have a U present. You do not have a T. So if you see in this image right here, anywhere you have an A, it's going to eventually, pair with the U. So only A and U go together. There's no T present. So this is the RNA at the bottom. You don't see any types of T's that are present. And finally, to kind of summarize what we've been talking about the entire time, a central dogma is basically just kind of a thought that you base a lot of other aspects on. So when we think about biology, we really focus on something called the central dogma of biology, which is DNA to RNA to protein. Real simple. I know this has a lot of words on it, but simplistically, it's DNA to RNA to protein. That's how everything works. We have a big message in our DNA. We pull out what we need. We make the RNA. That process is actually called transcription. We'll talk about it. And with that RNA, we take it from the RNA message and we translate it into a protein. And then eventually that protein goes out in our body and does some type of function. So all these things are interrelated. And I did want to make sure that we introduce that because we'll be talking about different aspects of the central dogma. And I want to make sure everyone's well aware of what that is. So as you see, this concludes our lecture, our recorded lecture at least. So again, if you watch this, this is really useful for you. And I would encourage you to, again, slow it down, speed it up, do whatever you need to do to make sure you get the material. And we will conclude with this one.